Hello everybody, greetings from Krakow, old town in Poland. I am from University of Science and Technology from Krakow, and if you agree I will tell you some maybe new information concerning biomineralization of lung cancer tumors. Biomineralization, this is mineralization of biological tissue. In Krakow, in my cathedral, in my department, we do investigation of various cancer, various tumors, and this lecture will concern only one type of cancer and special type of cancer of lungs. There are methods we use for the examination and equipment: stereo microscopy, polarizing light microscopy, scanning microscopy with equipment necessary for chemical analysis. For examination were prepared special thin sections and was selected one type of carcinoma. This is difficult for pronunciation for mineralogists. I am not oncologist, I am not medic, so you better read yourself. But these examinations were made together with of course, with oncologists. They prepare samples, and there is the macroscopic picture of a carcinoma, carcinoma planoepithelial. It's according to the classification is 1.31 number. It has this number. And this is way of the development of the cancer, and using the same pieces or parallel samples, we did examination using polarizing light microscopy and especially scanning microscopy, because scanning microscopy is very helpful when one examine mineralization. It's necessary to say that mineralization don't means grains or crystals, because mineralization, this type of mineralization observed in cancer or in lung cancer, this is mineralization, secret mineralization. What this means? This means that elements are substituted to the atomic structure of various tissue, and sometimes this secret mineralization, when it's growing, it develops, it starts to be changed to the mineralization we may observe under the microscopy, grains or crystals. But first of all, it's secret mineralization, and this substitution of elements into the biological structures lead to many problems, because tissue look like normal, but they are harder, they are not soft, they are not transparent, etc. So macroscopic picture means nothing. It's necessary to do chemical analysis and observations under higher magnifications. And there are structures devoted, fragments of tumors of lung cancer devoted for the examination. And now we have examples of chemical analysis done at many places of the same tumor. And you see, I will not read because everybody is able to read himself. And what is interesting, that even at the same tumor, at various parts we have various concentrations of elements. At one place we have phosphorus and sulfur and calcium, chlorium, and other place there is practically nothing, or there are present other elements. The question is very important because it's not, there is no explanation if first is mineralization of tissue and this leads to the evolution of cells and formation of the cancer, or cancer is producing some liquids, mineralized liquids, and because of this analysis show high concentration of elements. Probably is situation first, this means mineralization of human liquids lead to the formation of tumor, and the tumor is producing some substances mineralizing tissue, and there is not known a relation between the scale of the mineralization, primary mineralization, and this secondary mineralization by tumor, by cells of tumor. Independently, there are pictures of various places of lung cancer, the same all time. We try to collect collection of the same tumor from various patients, from various peoples. And we did many analyses. I show you only examples, not all what we did. So calcium at this place, and sulfur again, and natrium, and drawing showed the elements in one of places. And next, the same sample, and next place, practically only phosphorus, natrium, and sulfur, not too much. And it's drawing. And older men 59, the same carcinoma, the same cancer. This is histological picture. I could not explain because I am not specialist, but it was selected by oncologists for next analysis. And there are pictures of tumor taken under the scanning microscopy. This is the scale, 1 millimeter, and these points show places of chemical analysis. And what we have: calcium, iron, and again phosphorus, sulfur, and drawing of concentration. And as we observe together with friends, medics, it's often no relation between cancer and age. Generally, older persons are more often sick. They often have cancer, lung cancer, not only lung but generally. And in my opinion, I work in these problems last 30 years, maybe 35. Is connected with the general mineralization, so-called calcification of human organism. Our investigation show when develops osteoporosis, elements are not removed from the bones and removed from the organism next. They are circulating in the blood vessels and at places where are centers of crystallization start, these places, this tissue with centers start to be mineralized. So calcification develops sometime in heart, sometime in cartilage, sometime in other places. In one person is mineralized in the brain, second in blood vessels, etc. And this depends on centers of crystallization. And this mineralization of human liquids and blood lead to is the reason of general calcification of human body. And if liquids in organism are mineralized, these may lead to changes in the tissue, not only presence of elements, presence of crystals of grains, but this may lead to the changes of biological structures. And these changed structures work other way than normal. They are not normal, they are mineralized. So, for example, blood vessels are hard, heart valves are not soft, and this lead to the bad functioning of valves and generally blood vessel system, etc., etc. Many results of so-called calcification. Not very big, not well seen, but very dangerous. There are various biological structures of cancer taken under the consideration and examined by us. And example number 2, this man, 59 years old, and 0.3 in tissue in cancer. We have similar situation that calcium is of course everywhere, phosphorus is everywhere, but sulfur is very frequent in this tissue. Next example of the microscopic picture, histological, showing differentiation of the cancer tissue, how they are differentiated. And there are pictures of scanning made using scanning microscopy, and places where we did examination at many places of the same sample. And again, what we have, aluminium, for example, and phosphorus very high, sulfur very high, potassium very high, and no iron, for example, no calcium. In other places, at other places there were these elements, magnesium and sodium, there no. Next place of examination, the same sample number P2, place number 5, not very far from point number 4 or 3 and other concentrations. And next man, 67 years old, the same carcinoma, left lung, histological picture. This histological observations were the base for selection of the material for scanning microscopy examination, observation, chemical analysis, and so. And the imaginations are not very big. This is 1 millimeter, so not necessary to obtain very big magnifications. Phosphorus and sulfur, it's very small, is in percent, weight percent, 1.1, yes, very low amount. And next, it's number 3 sample. This man, 67 years old, and we have aluminium and calcium and zinc and sulfur, and so. And man 65, it's other type of cancer, adenocarcinoma pulmonis. We did examination of a few samples of this cancer. This is again histological picture, and this black are concentration of carbon. This is minor. This is it was minor, still alive. Minor, and in lungs he had concentrations of carbon because he worked in underground mine of black carbon. And the scanning microscopy picture, and again phosphorus and sulfur is repeated, sorry. And structures, various structures of cancer, and magnification is slightly bigger, it's 550 micrometers, and analyzes at various points calcium, zinc, and phosphorus and sulfur. And what is the result of this examination? Not only determination of amount of the elements, but these obtained data are the base for the let's say intellectual work. And because of obtained results, we suppose that the reason of the cancer is connected with the division of DNA. When the cells are divided, one cell is divided for 2 cells, and DNA is divided on 2 parts. One part is going to the one new cell, and second is going to the second cell, new formed cell. And the liquid where division takes place is mineralized, so contains high amount of various elements and various components leading to the cancer. For example, asbestos, for example, some chemical products, not only elements, but cancerogenic components. It's possible that at the place where is part of DNA devoted for the procreation, is possible that at these mineralized conditions will be mistake of DNA. And there is this fragment of DNA is relatively long, devoted for the procreation, and is possible mistake at many places of this fragment. And first of all, is many places of possible mistake, and is many various substances which may change DNA at this part of DNA. So we have mixture of various places and various cancerogenic elements. When we take these under the consideration, we obtain a lot of various cancers. And in opinion of my friends, medics, oncologists, they told me that during the life we have many phases, many stages of cancer in our organism, but organism is enough strong to fight with this, and cancer don't develop. He is reduced and the cells are dying. But at some moments when organism is not enough strong, when we have defect on this small distance of DNA devoted for the procreation, this cell, one cell, start to be procreated very fast. At one type of cancer very fast, in other types of cancer very slow. One type of cancer leads to this type of changes in the organism, other to other changes, and this is great variation of various types of substitution of cancerogenic substances to the various places of DNA. And this investigation we made confirmed that in cancer tissue there are higher concentrations of various elements, and at one place of the same tumor we have one group of elements, at other place of the same tumor we have other elements. So this may suggest that the cancer is not only the reason of the one type of mineralization of liquids in between the cells and in cells, and this is probably confirmation that first of all mineralization of environment where the cells are divided may lead to the formation of cancer. But if cancer is formed and procreation of cells is going, this these newly created cells may produce various concentration of various elements too. And this process is sometimes very fast, sometimes very slow. And of course, it's a beginning of examination of tumors using mineralogical methods like X-ray and Raman spectrometry and infrared spectrometry. But this is kind of investigations very, I think, in future helpful for oncologists, for patients, and have to be continued. We only show the methods and first results. We did examination of other cancers, cancer of skin, cancer of mouth, cancer of breast, etc. So it's not only examination of lungs. So thank you for listening, and all the best to you. Good health. For doctors is very important. They have to be healthy. Thank you very much, and maybe goodbye.